Welcome to homework.
Here the question is, draw the structure of each peptide, label the N-terminal and C-terminal amino acids and all amide bonds. So first peptide is given, that is A, and it is wall glue. These are the abbreviation which is indicating that it is valine is the first amino acid taken and second amino acid should be glutamic acid. So first of all, we have to draw the structure of valine. Now when we are making the peptide, always remember NH2 and COH should come in one line because they are forming the amide bond. So first valine, NH2, this is going to make N-terminal amino acid. So after that, the alpha carbon, which is having the hydrogen, and the side chain, whatever it is. In case of valine, it is this one, and after that COH group. So this is the structure of valine, and second is glutamic acid. So when you are making the structure of glutamic acid, also remember that CONH2 bond formation, how it can take place. So NH2 should be here, and alpha carbon, then hydrogen, and then COH group, and the side chain will be this one, which is having one COH group. So this is the side chain of glutamic acid, and by the combination of these two, one water molecule is removed, and amide bond is formed. So one hydrogen from here and one OH group from here is removed and amide bond is formed, and you are getting this peptide. That is the first one, wall glue peptide, and here this one is the N-terminal amino acid, and this should be the second one, that is C-terminal, where the COH group at the end is C-terminal amino acid, and here it is glutamic acid. And the amide bond is in between these two. This is the amide bond. This is in case of A. Now come to B. B is GLY his LEU. That means this is the peptide you have to make, and this is indicating that it is in this, the first amino acid is glycine, second amino acid is histidine, and third one is leucine. And by that the peptide which is formed will be this one, and glycine structure should be, again when you are making the structure, make it in draw it in such a way so that you can make the amide bond easily. So one hydrogen here and one hydrogen here, which is not taking part in amide bond formation, and COH group. So this is glycine, first structure. Second structure should be, again NH2 group should be this side of histidine, and one alpha hydrogen and one COH group, and here it should be CH2 group, which is further attached to one ring in which one nitrogen is present, one double bond here, single bond, one NH bond here, and one double bond must be here. So this is one ring which is attached to R group of the histidine, and by that histidine molecule is formed. And third is leucine amino acid. In that again you are keeping the NH2 group this side, and alpha carbon hydrogen, and after that the carboxylic acid group, and this R group here in case of his leucine is CH2, which is directly attached to this whole group. This is leucine. Now what happens here again, one hydrogen from here and OH group from the COH part is removed, and by this water molecule is removed, and they are joining together and forming the amide bond. Here also OH group and hydrogen is removed and water molecule is going out, and by that these two amino acids are joined together and amide bond is formed. So by joining of these three, we are getting the this second peptide, and that is glyhisleu. And here also we have to remember that the group terminal which is containing NH2 group is actually N-terminal amino acid, and this N-terminal amino acid is glycine here, and C-terminal amino acid is leucine here, and these are the amide bonds which are formed by joining of two amino acids. Now come to the last one. Third is MATT. That means the amino acids are taken for the preparation of this peptide bond or peptide molecule is actually M is representing first amino acid, that is methionine. Methionine. This is the first amino acid. Second amino acid taken is alanine. A is representing alanine. And third one is threonine, that is THREONINE, and this is represented by T, abbreviation is T for this amino acid, and last one is again same, threonine. So it is also threonine. Now make the structure of this, and remember, for making the peptide bond you should have the amide, that is COOH group and NH2 group in one line, and all other side chains should be separate. So first molecule is N-terminal amino acid methionine, and here it is NH2 CH COOH, and the R group is here CH2 CH2 and SCH3. This is the R group present in methionine. This is joining with alanine. Alanine should be again NH2 group this side, then alpha carbon, then hydrogen, then COH group, and here it should be methyl group if the amino acid is alanine. Third molecule is threonine. Here again NH2 group should be this side, and alpha carbon and hydrogen and COH group. And the R group here is one carbon which is joining with the methyl group one side, OH group another side, and hydrogen at another side. Plus again NH2 group, same compound, same amino acid, C, H, C double bond O, OH is the last molecule, is same molecule, and here also one is methyl group, one is OH group, and one should be hydrogen. So this is the R group, and here again everywhere OH and hydrogen is removed and amide is formed, amide bond is formed, and by that they are joining together. So removal of this water molecule and joining of the molecule everywhere. It is easy when all are in one line. So removed, and by that all the four amino acids are joined together and peptide bonds are formed, amide bonds are formed. So this is the structure of the last peptide, and this is again methionine is the N-terminal amino acid, and here it is the C-terminal amino acid. One threonine molecule is C-terminal amino acid, and these are all amide bonds which are in between the amino acid, and by that they are joined together.